everybody. A very, very warm welcome to tonight's webinar. I'm really pleased to have you all with us, along with our distinguished guests for today. So tonight we're going to be talking about biomarkers. And some of you joining today might be thinking, what is a biomarker? And the full term is biological marker. And we're talking about things like blood tests, urine tests, stool tests, and where you're looking for a marker that tells us if someone has a medical condition and what that condition is doing. So, for example, many of you may be familiar with tests for diabetes and checking blood sugar. And this is really important for cavernoma because currently with cavernoma, we can't predict if or when somebody will have a bleed, and we'd really like to do that. Now, today's speakers are going to be talking about research that's in a laboratory, but our hope is that in time that will come to clinic, and we'd love to see there being tests that you could have as a patient to find out what your cavernoma is doing. So I'm just going to begin by introducing our speakers, and then I'm going to hand over to Peetra Magnusson, who will chair the presentations. So let me begin with Associate Professor Magnusson, who is Associate Professor, Group Leader and Director of the Rudbeck Laboratory at Uppsala University in Sweden. And she's been passionate about cavernomas for many years. We also have Professor Roberto Latini, who's joining us from Italy, who's the head of the Cardiovascular Medicine Unit at the Mario Negri Institute for Pharmacological Research. And some of you may know his work in respect of his propranolol trials. We also have Dr. Francesca Lazzaroni, who works with Roberta, and she's a postdoctorate researcher at the Ospedale Policlinico di Milano in Italy. And please forgive my very, very poor Italian saying of that. And we also have Favour Onyeogaziri, who works with Peetra and is also based in Sweden. And she's a PhD student in neurovascular biology, specialising in CCM. So thank you so much for giving up your evening to talk to our members today. We've had 54 people registered. Many of them have already joined us. So I will now hand over to Peetra. Thank you, Peetra.
Thank you so much, Helen, and thanks for arranging this webinar. It's a pleasure for us all to be here, and especially really exciting to have these two presentations. And Roberto will, in the end of this session, give us some really new exciting data that's not published, because what we will show today is work that we've been working on for a long time, since the trial has been going on for 2 years, and then it was ended. Samples were analyzed, and we have been working with that. And Francesca has done a great job on the circulating biomarkers. We put together the paper that was accepted for publication and online early this year. And in parallel with that, we have actually in Uppsala also looked at biomarkers, but then focally in the brain, looking at bio samples from the brain, biopsies. And that has been a possibility to actually really look what's going on in the brain, while Francesca has looked at what's going on in the circulation coming out from the brain. And this will hopefully guide us to understand what is going on and when is it happening, and what is the difference from what goes out into the circulation and what is happening inside the brain. So it's going to be really exciting to hear both Francesca and Favour to present, and then the new exciting things from Roberto regarding the microbiota, which is also a very hot topic in the CCM field. So it's really a pleasure to be here tonight, and I hope you will have a wonderful time here now with the presentations coming up. And I turn to Francesca and ask you to take the word and lead from here.
So good evening to everyone, and thanks a lot for this opportunity. And today I'm really glad to present you our research and our project focused on the biomarker assessment in the TREAT-CCM clinical trial. So first of all, just a couple of words regarding the CCM treat clinical trial. So in the first part of the talk, I would try to explain the main features of the clinical trial and the TREAT-CCM clinical aims from the clinical point of view, but also from the biological point of view. And in the second part of the talk, I will focus the attention on my side, so the TREAT-CCM from the bench, and then regarding the circulating biomarkers and protein biomarkers with the clinical outcome from the research. So just a couple of words regarding the the link between the cerebral cavern mass formation and the propranolol, because today the only curative treatment of cavernomas is the surgical correction when feasible, depending on the location of the lesions. And so far, no drugs to cure or slow down the progression of CCM are available. So propranolol is a non-selective beta blocker, widely used for the treatment of hypertension, migraine, and essential tremor. And it as well very well described that propranolol as well recognizes safety. So propranolol has not been found to be effective in the treatment of infantile cutaneous hemangioma, and there are some anecdotal reports that have been published on propranolol efficacy in CCM in the absence of matched controls. So speaking about the TREAT-CCM clinical trial, we had clinical aims. So the purpose of this exploratory trial is to assess the effect of the propranolol in reducing the burden of cerebrovascular lesions and clinical events in patients with CCM. But on the other side, we have also molecular aims, so to characterize the circulating biomarkers starting from the peripheral blood to identify a specific signature in CCM patients in comparison to healthy controls. We aimed to identify some plasma protein biomarkers of CCM patients to assess the severity of the disease and predict its progression. And the story started also to study the effect of propranolol. So this is just a snapshot of our study with the sampling of clinical parameters and biological specimens. So we were able to collect the clinical parameters, but also the biological samples from patients at the 3 different time points, the randomization after 1 year and after 2 months. And the clinical trial was ended at the end of the October 2021. And the paper was published thanks to Roberto Latini and Silvia Lanfranconi at the end of the year 2022. So the biological point of view of the TREAT-CCM. So starting from the clinical aspects, we tried to define the effects of the propranolol treatment on the cerebral cavernous lesions. But on the other hand, we aimed also to use the peripheral blood and biological samples to identify plasma biomarkers to assess the severity of the disease and predict its progression. So we were able to collect and to study the same cohort of patients that we have defined in the TREAT-CCM clinical trial, and we have already published 71 patients affected by familial CCM and 17 healthy controls. We have collected the biological samples, such as blood, and also the stool samples, and Dr. Latini will explain the project regarding the relationship between gut and microbiome in the next presentations. And starting from the blood samples, we were able to study the circulating microRNAs, so the small molecules, the small transcripts that are free in the peripheral blood and that are linked in the literature with many cancers and many cardiovascular diseases. On the other hand, we focus the attention on the circulating biomarkers, so the protein side of the biomarkers. So the protein aspect, we have already published at last January, so in 2024. And our workflow, so our approach was based on 4 different molecular approaches in the lab. So we defined a pool of molecules that are already published in the literature because they are already linked and described as associated with some vascular disease and the cerebral cavernous malformation. So we tried to define and to study these biomarkers, but also we studied and we started with a non-targeted project, so non-targeted discovery of new proteins that maybe can be associated with the familial cerebral cavernous malformation in collaboration with the Peetra Magnusson at Uppsala University and with the Olink company. So in this way, we were able to discover other proteins that are linked to the CCM disease and vascular disease. And here we tried to connect all these biomarkers with some biochemical markers that are commonly studied and that are commonly defined in the peripheral blood of patients, such as cholesterol, CRP, vitamin D, and also the pentraxin 3. And finally, we tried to confirm our data, also using another approach to study 4 molecules that are related to the vascular malformations. So what we have shown is that to gain insight into the proteome landscape of CCM, we used by the high throughput automated ELISA assays, we were able to study the plasma proteome landscape, and we don't find any type of differences, and we not define the effect of propranolol treatment on the biomarker concentration after 2 years of follow-up. And in this way, we were able to study the cohort of patients as a single study. But so far, we have identified that we have highlighted that the familial CCM patients have higher levels of circulating biomarkers that are associated with inflammation and angiogenesis. Here you can see just a snapshot regarding different molecules already related to inflammation and angiogenesis. So using a combination of these biomarkers, we were able to define that the proteomic landscape of peripheral blood of patients with the familial CCM are characterized by inflammatory biomarkers, but also using these biomarkers, we were able to genotype patients. And here we can see that the patients affected by the CCM3 disease are as more inflamed, so with the high expression of these inflammatory molecules. But thanks to a non-targeted exploratory approach to define proteins related to the inflammation, we were able to define that the blood levels of ROBO4, thrombomodulin, and CRP, so 3 fundamental biomarkers related to the inflammation, can predict the clinical adverse events. And then we were able to predict disease progression and severity. And we considered the occurrence of the new symptomatic intracerebral hemorrhages or focal neural deficits as the primary outcome. And here just different protein biomarkers related to the inflammation, able to predict the formation also of new MRI detectable lesions during the 2-year follow-up of the clinical trial. So using a combination of these protein biomarkers, such as FLT3 ligand, CDCP1, and FGF21, we were able to show the statistically significant expression for patients developing at least 5 new lesions. So the take-home messages of these research project is that the high blood levels of these circulating biomarkers pinpoint that the marker of the inflammatory and pro-angiogenic activation that contributes to the onset and progression of CCM has a strong prognostic and diagnostic value. So the potential of biomarker screening starting from peripheral blood in CCM was further exemplified by our discovery that disease progression in patients with clinical lesion activity during the 2 years follow-up was predictable based on blood levels of proteins involved in vascular stability during angiogenesis and inflammation processes. And as the last thing that is ongoing now, and then we aim to publish as soon as possible, we started also to study not only the proteomic landscape, but also the transcriptomic landscape of the blood coming from patients. And speaking about the transcriptomic point of view, we studied the microRNAs that are the very short molecules, so 25 nucleotides in length, that are free to circulate in blood coming from healthy controls and coming from patients. And what we have done a couple of years ago is to start to sequence the microRNA landscape of 22 CCM patients matched with 21 healthy controls. And we define a very small pool of 36 microRNA that are differentially expressed between CCM patients and healthy controls. So again, we define another type of difference between patients and healthy controls. And again, the keywords of this exploratory approach was the inflammation and angiogenesis, because all of these microRNAs, as well as the proteins that we have already shown, are related to the angiogenesis, inflammation, cell migration, and also the endothelial cell remodeling. And at this moment, so with Roberto, we are trying to define a sort of the landscape and also the functional activity of these microRNA, trying to relate the microRNA function and the proteomic landscape. And the last step will be explained by Roberto, and it will be related to the microbiome and the interesting link between gut and microbiome in the CCM patients. So this is the last slide, also to introduce the next work that we have to do in a few months. And I would like to say thank you to all of you for the attention and to all of the collaborators that have worked on the proteomic and transcriptomic landscape, and all the collaborators, and the main special thanks to Elisabetta Deiana, Roberto Latini, and Peetra Magnusson.
Thank you very much, Francesca. Lovely presentation. And we will actually move on to the next speaker, which is Favour.
Good evening, everyone. Thank you for being here. My name is Favour. I'm a PhD student in Peetra Magnusson's group. And for the next few minutes, I will be sharing with you the work we've done in this past few years, looking at proteomics and human CCMs and how this has revealed new biomarkers involved in neurovascular dysfunction. Just here, this is a QR code that links you to the recently published paper. I would also pop this up again at the end of my presentation. So let's get to it. When we think about CCM, typically we think about the endothelial cells, and this is because when patients have CCM, then it's usually the endothelial cells that become enlarged and dilated, forming this popcorn-like lesions. But we have to think of the fact that the endothelial cells do not exist in isolation. They actually are part of this system in the brain called the neurovascular unit, meaning that you have the endothelial cells, you have cells that support them, you have cells like the pericytes and astrocytes, you also have neurons and other cells, and so all together, these form like the functioning unit of the brain. And so I put this to you because then we would see as we go on that CCM is not just about the endothelial cells, but that everything is affected. So last week I went on PubMed and I did a quick search on cerebral cavernous malformations, and more than 2,700 articles have been published on this, which means that there is a whole lot that we now know about CCM since the 1960s. But the truth is that there's still so much we do not know. And so we want to better understand the molecular mechanisms of CCM, because if we understand what causes the disease, then maybe we are able to find therapies that can help it. And we have lots of animal models and we have lots of cell models. They are great, but they are not perfect. In addition, we also would like to have biomarkers that can predict the disease status or the disease severity, because then this translated to the clinics would help doctors monitor patients better. And so to do this, we decided to do a multiplex assay. The advantage of this Olink assay was the fact that it required low sample volume and that we could also do it with brain biopsies. Once in a while, we have associations with surgeons, and when they do surgeries, they are able to sometimes give us these biopsies. And these are precious. We like to use them for as much as we can. So this was the perfect assay, since we could use very little volumes or amounts of these biopsies to do this. The other advantage of the Olink assay is that it allows us look for multiple biomarkers. In this study, we looked at more than 180 proteins. And so quickly, what we did was we took these biopsies from patients that had undergone surgery. We had 7 patients in total and 3 healthy or control samples. And one of them, this patient 6, was actually a familial patient, while the other ones were sporadics. We took these, we were able to isolate proteins from these fresh frozen samples, and then we sent it for analysis, where with sensitive methods, they were able to detect differences if there were in this 184 biomarkers that we had checked for. And so they sent us the results after that. And that's what we would share with you today. So after the analysis, they are able to show us what proteins are differentially expressed, meaning are different in the controls compared to the CCMs. And so here in this graph, in blue are the proteins that were downregulated, meaning that they were reduced in the CCM samples compared to the healthy controls. In red are the samples that were increased, or rather the proteins that were increased in CCMs compared to controls. And in gray are the ones where there were no differences. So let's start with the downregulated proteins. We found only 5 proteins that were downregulated, like I said, reduced in the CCMs. And when we looked at what their function is, we realized that these are highly expressed in neurons and other cell types in the brain, indicating that they are necessary for the normal performance of the neural cells. We went to our animal model because that's what we often use to sort of model CCM. And we saw that, interestingly, this protein here, contactin 1, was also downregulated in our mice. And so that was like a further confirmation of this protein. We went to the literature and we realized that contactin 1 is involved in the myelination of axons, which means that if it's not there, then it would actually affect neuronal function. And based on this, we think that this could be one of the reasons why CCM patients experience neural deficits, because if any of these proteins that are involved or necessary for neural health are reduced, then that could lead to some of the symptoms patients face. Now, going on, the upregulated proteins, so the proteins that are increased in CCMs, these were a whole lot. And in red here, these are the ones that we also saw in our mice model, which further validates this method. And so for the rest of my presentation, I will take you through how we validated some of these and the conclusions we made. Let's start by endothelial activation. The endothelial cells line the lumens, and when they are quiescent or when everything is fine, then you have maybe some immune cells and blood cells, everything passing through the vessels. And the endothelial cells themselves have these so-called adhesion molecules, but you don't have so many of them being expressed. But when there is a trigger or a stimuli, such as when inflammation happens, then it also activates these endothelial cells. And so suddenly they begin to upregulate more adhesion molecules. And one function that these molecules do is that it allows the binding of immune cells, here just shown with these. So these can bind endothelial cells, extravasate, and do what they need to do. Now, when we looked at the proteins that had popped up in our assay, one that was very interesting was CD93. Because not only is it an adhesion molecule, according to the literature, it's also involved in so many other things. It functions in angiogenesis, so the growth or making of new endothelial cells, but it's also involved in the migration, so the movement of these endothelial cells. But it also functions in regulating immune function. And so we thought, wow, this is a nice protein to further validate. And that's what we did. We went to the biopsies that we had used for these study, and we stained for CD93. So here in magenta is CD93, and in green is von Willebrand factor, which just labels all the endothelial cells. And what we could see was definitely, when we compare the controls to the CCMs, that there was definitely an increase in CD93. We went to our mouse model and we also validated this. So here to the right, the top is the wild type, meaning the control or the healthy mice. And at the bottom here are mice that we've induced to have CCM. And once again, you can see this increase in CD93. And here we plot this graph. And so this tells us that definitely endothelial cells are becoming more activated. This was great. So we went on to validate some other things. Now I will tell you about the extracellular matrix. When we think of the endothelial cells, we have to think of the fact that there is this supporting layer behind the endothelial cells that's needed for it to function properly. So in the extracellular matrix or the basal lamina, we have supporting cells, we have matrix that's needed for the connection of the endothelial cells to other cell types and to also promote their growth and their function. And so you can imagine that if anything is dysregulated here, it would also affect the endothelial cells. Now, this popped up in our study. So we saw that there seemed to be disassembly of the endothelial cell matrix. And so we decided to validate this further. Here, we used alpha SMA to validate this. And here in the control or the healthy donor biopsies, you can see what should happen or what it should be like. In magenta, once again, you see the supporting cells sort of around the vessels, supporting them. But here at the bottom, this is what the CCM patient looked like, that this was definitely dysregulated and nothing like what the control samples should be. Now, based on our studies, we thought these could actually be fibroblasts, which is a type of the supporting cells that are in the basal lamina, and maybe they become activated and have this, phenotype or characteristic. And so we went to the literature, and interestingly, a paper came out in the time when we were publishing this, really showing this same phenotype, right? And here they were able to really show that these were fibroblasts. And so based on this, we know that a different cell type called fibroblasts are also activated and being dysregulated in CCM. Now, moving forward, another thing that popped up in our analysis was neuroinflammation. This is a word that essentially means inflammation of cell types, other cell types in the brain. And so here, for example, the astrocytes can become activated, or microglia cells can become activated. And so this term neuroinflammation is a broad term that covers all of these. when we looked at our results from the O-link assay, we could see that genes involving microglia and astrocyte migration seemed to be upregulated in the CCM samples. And so we decided to validate this using GFAP. Now, this is a marker expressed by all astrocytes. And here you can see what the control biopsies look like. Essentially, they are like these star-shaped cells, all happy and nice. But when astrocytes become activated, then they lose this star shape and instead just form a totally different shape, as you can see here. And this is what we saw in the CCM patients, that these cell types, astrocytes, definitely become more activated when we compare them to the controls. Now, other studies by other labs have shown that when this happens, this can sort of promote hypoxia in the cells. And once again, this could maybe lead to, some of the symptoms that patients face. And so with this study, we are sort of seeing things that we knew before, but also new things and putting them, all together to learn amazing things happening in CCM. We've talked about the astrocytes, but we also wanted to look at the microglial cells. Now, microglial cells have a special function that they work as resident immune cells, but usually they should not be activated. They should be there. They would be able to take up debris or dead cells in the brain and continue their function. But here, when we compared what they looked like in the control samples compared to the CCM, then you can imagine here we use IBA1 as a marker of these microglial cells, and you can definitely see and appreciate this marked increase in IBA1 in the CCM patient samples compared to the controls. Now, we looked at the literature and we saw that actually fibrinogen, which is an end product that happens during blood clots or when thrombi form, has been shown in some other diseases to be toxic to microglial cells. And this is it. Here in magenta, we have fibrinogen. And in the control patients, you can appreciate that there is no co-localization, or it doesn't meet the IBA1 positive cells. But take a look at the CCM patients. Here, co-localization shows up in white, and you can definitely appreciate that there is more of this happening compared to the control samples. And so what we see now is that while we know from different studies that clots or thrombi can cause hypoxia and can occlude vessels, but also here we are seeing that they can also have other negative effects on the cells. And so this is why, once again, we believe that targeting thrombi might be a good strategy in CCM. Now, going on, in our samples, we had 3 patients that had ruptures in their lesions, and then 3 others that were not ruptured. And we were able to do analysis on these. We separated them and we checked for differences in these. And indeed, we had more than 10 proteins that were either increased or decreased in the ruptured compared to the ruptured samples. And without going into so much detail here, what we just want to point out is that we believe that instead of us as a field focusing or trying to find the one protein that would be the biomarker for CCM, that instead we might look at a profile of markers. For example, here we have 3 markers that were reduced in ruptured cases, while here we have more than 10 markers increased in ruptured cases. And so perhaps a biomarker profile like this, where you have some proteins being increased and others being decreased, might be beneficial for CCM. And here I would just like to point out that we actually saw that CDCP1 was increased in the ruptured samples compared to the non-ruptured samples. And this was one of the markers that Francesca and colleagues had identified in the Treat CCM familial patient plasma study that Francesca just talked to us about. And so this was really interesting to see. Now, going on up until now, I've told you that the endothelial cells become activated, the extracellular matrix or the supporting cells around the endothelial cells also become dysregulated, but also other cell types in the brain are being changed. But what does this mean? You can imagine that when you have triggers of dysregulation or when the body feels like something is wrong, it sends stimuli or triggers the immune cells, and this can cause them to be activated. And this is something we already know, that in CCM, there is increased inflammation. And we could also see this in our panel. Here, you can see the control cells in the left panel, and you can appreciate that there is barely anything here. But here to the right, you can see that in 3 or in different CCM patients, then we could see different kinds of immune cells present in these samples. And so we know definitely that it's not just one kind of immune cell that's being changed or dysregulated, but multiple cells are present in the CCMs. This was interesting, and so we wanted to understand this further. And so we decided to use a marker that is expressed by all immune cells. This is called CD45, and here in magenta. And CD31 is a marker that lines the vessels or the lesions. And you can see clearly here in the control samples, there is essentially no CD45 cell here, meaning there are no immune cells. But then let's take a look at the patient samples. You can see here in this patient, these are the lesions, right? The enlarged vessels, and definitely we could see immune cells inside them. But then also, when we looked outside of the lesions, we could also see immune cells, meaning that the immune cells had extravasated or had left the vessels and gone into the tissue. Now, this was expected, but what was unexpected was the fact that when we looked at regions in the tissue that were far away from the lesions, we could still see a huge accumulation of these immune cells. And so this tells us that the immune cells are here, they are activated, they come in and they extravasate deep within the tissue. Now, what does this mean and why is this important, you might ask? That's a good question. One of the things we wanted to find out was how the border and the lesions might be different from control brains. Without going into so much details, we know that the control brains or the control biopsies are healthy. And let's imagine that the lesions are not healthy, right? And so let's say they are here. Now, when we looked at what was happening in the border of the lesion areas, we actually saw that the border area was not as healthy as the control. It was not as bad as the lesion, but it was not as healthy, meaning that the lesions were affecting the borders and making changes there. And so we asked ourselves, how could this be happening? And we believe it is because of this. Now, let me tell you why.
Favour, we have to wrap up rather soon because it's already 8:45.
Okay, all right. And
Sorry.
so we checked for these, and actually we could see that it's these immune cells that are extravasating out that are causing these changes in the surrounding brain border regions. And so if we can stop this, then perhaps we can limit these changes. And so just to conclude, I hope I've been able to convince you or tell you the fact that now when we look at CCM, we need to look beyond the endothelial cells and realize that in CCM, all the other cell types are being activated or changed. And during a rupture, this just gets worse. And so these are some of the things that we would continue to look at in future. And I would stop here, and I thank you for listening. I thank Peetra, I thank Suvi for heading up this project, and all our collaborators, funders, the patients and their families. Thank you so much.
Thanks a lot, Favour, for going through this so nicely and thoroughly. Excellent. So Roberto, please tell us the latest news on the
Peetra.
microbiota.
Thank you, Helen. I am honored to be here to present you a few slides of some preliminary work. I'm happy that Jana, with whom I have been knowing for many years, is present also. Hello, Jana. So nothing new. I am from Mario Negri, and it's a bit kind of an ugly gray building in Milano that Peetra knows because she visited us. Where does this story start? Actually, the story starts from the paper, it was, I think, 2016, the paper by Alan Tang, who is the fourth author in this more recent paper in humans, and he studied in murine models of CCM the role of microbiome. In extreme synthesis, he demonstrated somewhat that the gram-negative bacteria were pouring into the bloodstream LPS, and this LPS, through interaction with toll-like receptor 4, was leading to increased permeability, which is a major alteration seen in CCM. And Tang and his collaborators were also able to revert the disease by playing with modulating the microbiome. In this 2020 paper in Nature Communication, the authors, pretty much the same group that is centered over Isama Vaden, his group in Chicago for the clinical work, they found out in a series of patients with CCM, please note that they are not necessarily familial CCM, but are cerebral cavernous malformations that have bled within the 6 months before the observation. They found 3 common species, as they write, by combining multivariate machine learning-based random forest analysis. So, unfortunately, I cannot explain you really the meaning of all this, but it looks like they use sophisticated method, and they found the gram-negative bacteria that was increased, and 2 gram-positive bacteria that were decreased. The increase in gram-negative bacteria may suggest an increase in LPS in the bloodstream, sort of similar to what was observed in the murine model. And this is the relative abundance of these bacteria. But if you look carefully, the abundance of these bacteria is really rather low, mostly far below 5%. Nonetheless, they were able to, combining the 3 bacteria, to obtain a ROC, so a curve that sort of shows you how predictive are these 3 bacteria, of 0.8280. Well, the perfect curve would be 1, but this gets very close, so it's rather encouraging. So what have we done? Back in 2016, when we started the trial, the scheme is here, and already Francesca showed. The centers were distributed over Italy. We span over the different latitudes to be politically correct and representative, but and we collected stool samples at baseline randomization and after 2 years. This is a long table, and I don't expect you read it because we would be here until midnight, but this is just to tell you that the proportion of patients that had functional neurological deficits as a clinical history or intracerebral hemorrhage was distributed unevenly, in particular functional neurological deficits that were twice as much more frequent in patients that didn't provide stools. So this is a limitation of the study itself, and we have to acknowledge it. And these are the preliminary results of an analysis performed on the data that were obtained in Luigi Nesi lab in Institute, European Institute of Oncology. I will show you very preliminary data, but this is the alpha diversity assessment that sort of tells you the different species represented in population of bacteria in these stool samples from patients of the CCM versus healthy volunteers. And you can see that there is no statistical significance. But if we look at the beta diversity, just again trying to capture if there are differences between patients, healthy controls, 17 and 31 CCM patients, the P, you don't need to have in-depth knowledge of statistics, is far from being statistically significant. So the results to date, and I don't go more in depth into these results because now we are identifying the individual bacterial species, but the results here are not really going along the conclusion by the paper, the US paper. In the US paper, they claim that weighted combination of microbiome signatures and plasma inflammatory biomarkers enhance association with disease severity and hemorrhage. Is really a sentence that is rather difficult to be interpreted and literally translated, but we considered our results that Francesca presented, and we asked ourselves, what about the inflammatory biomarkers, circulating biomarkers? And here you can see that we had a remarkable difference in CD14, for example, that is an antigen surface antigen of monocytes, being about twofold increase in patients, this in blood, and also LBP, like a polysaccharide binding protein, that was about twofold increase in diseased patients. Unfortunately, we cannot see any major difference in the microbiome. And to complete the picture, we also found, as Francesca showed, through Olink proteomics, that the GDF15 and many other molecules, sort of markers, readouts of increased activation of inflammation were increased in blood, but the same picture could not be seen in terms of bacterial population in the microbiome. I would stop here, and I hope soon to provide you and hopefully the scientific community with more definitive data. I want to thank Teresa Ravizza in Mario Negri Institute, who helped us a lot, and Ray Sahin, who is the bioinformatician who performed the analysis, and then the people from the group at Mario Negri, Francesca Lazzaroni, Luigi Nesi, and Carlotta Catozzi, and Angeli Marcandon, who did the microbiome 16S assays. I thank you for your attention, and I'm open to, I would say, to suggestions, because whenever you hit as a researcher a negative, say, neutral finding, this is very difficult to be supported by evidence and presented. So here we are. Thanks for your attention, and honored to be to have been made able to present. Thank you.
Thank you very much, Roberto. I can agree that it's always complicated when you don't have low significant numbers and great changes. But still, there is a value in the findings that is collected. So well done work there. Helen, now it's time for some questions. And
fantastic. Thank you so much, everybody. Those were just fascinating presentations. And the thing that excites me is how much more we're learning about cavernomas, and that goal of at some point having a test in clinic so people can have some warning as to when they might have a bleed or when their condition's going to progress. So thank you ever so much. So if you haven't already, as Petra said, please put your questions in the Q&A. I'll read those out and we'll give an opportunity for the panel to respond. We're at 8 o'clock British summertime. So I'd suggest we allow another 15 minutes and then we'll wrap up. And we've still got really good, healthy numbers in terms of the participants who first joined. So thank you ever so much. And just a reminder, this will be recorded. It will be on our YouTube channel. So please do subscribe if you haven't already. I know I want to go back and watch it all over again because there's a lot there to digest. If you're not familiar with the scientific terms, you may want to go back and rewatch, but I certainly took away a lot. So just one question I'm going to begin with, a very quick summarizing question is, because I know this will be on people's minds, how long do you think it might be until we potentially have a test in clinic that patients could have to help predict when symptoms may occur? And I know that's a bit, how long is a piece of string? Roberto, go ahead.
Well, if I can transfer from the cardiovascular disease area where there are several blood tests. Well, take one example, troponin for myocardial infarction. To develop a reliable test, even with much larger numbers than these ones, because as Petra correctly pointed out, 31 disease patients are not that many, but we have to deal with the rare disease. So it's going to be a long way. Technologies are very much developed, but for example, to develop natriuretic peptides as biomarkers of outcome in cardiac failure, it took about 40 years. So now we have more developed technologies, but consider that we need more work. But let me conclude, we need more work done by independent groups, which means, well, this is true for any scientific research, but we need to have confirmatory works or contradictory works from different independent groups, not just one single group providing the truth, which is unacceptable. I would never sell this truth to patients. I respect them too much. So I think the efforts also in Europe should be really fostered and encouraged a lot. And I'm happy to be here because there are no privileges. We have to really share resources. And Petra knows how hard we tried. We still try. So
yes,
we may have some good news in the near future.
And Roberto, just to say, we certainly believe this at Cavernoma Alliance UK as well, how important this research is. And I know the European Cavernoma Alliance feel that as well. And as a charity, we're going to do all we can to keep promoting research like this, because you're absolutely right. The more studies, the better. It's a rare condition. It takes longer for us to get these tests because of the numbers of people available. So we're thrilled, though, that you're here today helping to raise that awareness. And hopefully we can get some scientists watching this webinar afterwards. And we want those PhD students looking for a new title to think, this is what I want to focus on, like Favour think. I want to put my PhD study on that. So I'm now going to go through the questions that have been asked, and I'll take these in order. So the first one, given there is an association with angiogenesis, inflammation, etc. in familial cavernoma, would you expect patients to have other risks for comorbidities elsewhere in the body, for example, systemic inflammatory conditions, or does it only relate to the brain? And I just want to say a huge thank you to you all for working on cavernomas. I'm blown away by how fantastic your work is and what groundbreaking, life-changing work you've done. As patients, it makes us feel hope for the future it can feel a bit dark at sometimes. Thank you for helping us. Oh, isn't that a lovely?
Oh, that was wonderful.
Oh, that's very, very sweet. So who would like to take that question? And I'll do one person answering per question just because of time. So who'd like to take that one?
Well, if I can just comment, because yes, you don't want a big inflammation going on in your whole body, and Francesca showed data on inflammatory markers. We also must pinpoint that the tests that have been done and used are usually very sensitive. So it has also to be compared to what does that mean with a severe inflammation. And we have now then also investigated locally in the brain. So that is, of course, more direct to see what's going on in the brain. And then it's spreading into the body. And that would, of course, be far lower levels than if you have a full inflammation going on centrally in your body. And Roberto, if you would like to add on.
Following your sentence is very reasonable. In fact, in our 71 patients on which Francesca and you performed the assays, there were no inflammatory disease. We specifically searched for it.
Yeah.
And I think 2 or 3 had mild diabetes, no cardiovascular disease. And again, they were 45-year-old in age, so let's call them healthy. Besides again, we observed statistically significant but very limited increase in inflammatory biomarkers. Peetra is right. If there were inflammatory diseases like, for example, COPD, chronic obstructive lung disease, or infection, you would have skyrocketing levels, so which are absolutely would be out of the range we are observing here. So it looks like there are inflammatory markers leaking into from the brain into the bloodstream that just increase very little in concentration. And that makes more difficult the work, but I would exclude generalized inflammation. But it's a very good point. We should look more carefully, probably. Thank you.
Thank you. Thank you. That was a really comprehensive answer. So the next question, also linked to inflammation. Can subtle changes of neuroinflammation, brilliantly described by Favour, cause progression of neuropathic symptoms despite the lesion looking quiescent on MR scan? I have a partially resected C3, C4 spinal cavernoma. So is there anything more you'd like to say on that point?
Favour.
Yes, thank you for the question. So right now it's still definitely a hypothesis, but based on our studies, we think yes, because you can imagine that when there's dysregulation or when these cell types that should be healthy become unhealthy, then they begin to release things, right, and signal to neurons and other cell types in the brain, which can also affect their function. So we believe it's a definite possibility, and that's something we've now, or we plan to in the next few months, we want to start looking into that. And so hopefully we would be able to have a more concrete answer. But right now we think it's a high possibility that could happen. So
thank you very much, Favour. So the next question is there anything any of those of us who have cavernoma can do to help research? Just as a representative of patient advocacy group, I'd say 2 key things. One is when research trials take place, sign up. As a rare condition, it's really hard for researchers to get enough patients. So when those trials happen, we will certainly let you know. In the UK, we've recently had the CARE trial, and the results of that trial were published last week, which is really exciting. And the second one is a plug fundraising. We recently, as a charity, have funded the start of a proof of concept research with UCL here in the UK on a gene therapy. And I'm really pleased to say they're working with Peetra's team. It's a project in partnership, which just shows the value of that European collaboration. So that fundraising also can really help us then to have the money to help take these projects forward. And Favour, you have your hand raised.
Yes, I just wanted to say something that I and Petra always talk about, the fact that in the ideal world, we would want matched samples. So, for example, when patients go into surgery, and so if it's possible to donate your biopsies to science, then we appreciate it because patient materials are so precious. And so I understand that when you're going into surgery, that's not what you're thinking about. But if that option comes up, then it's always, it's always great to do. And then also blood samples and all of those are great. So ask your physicians to say what's the possibility. And if you're asked, please say yes. Thank you.
And
Thank you.
stay as curious as you are and engaged. I think that's also because it's motivating us a lot.
Oh, thank you ever so much. And just also a plug for the patient registry of Alliance to Cure. They're our US counterparts. We don't have a patient registry currently in Europe, but in the US they have one. You can go onto their website, Alliance to Cure, and sign up there. And that again provides really important data for researchers to tap into. And thank you, Roberto, for acknowledging Professor Rustam Salman Al-Shahi. He's the chair of CAUK's Medical Advisory Board and is doing lots of incredibly important work in the research field. So the next question. My serum triglycerides were 4.9 in November and 1.5 3 months later. Could this be connected to a bleed? Who'd like to answer that question?
The clinician.
Roberto.
Yeah,
Roberto,
please.
No, no idea.
No.
I don't remember, but maybe Francesca has the data at hand. What was the difference between patients and CCM patients and healthy volunteers in triglycerides? The only thing that I know is that triglyceride plasma concentrations are highly sensitive to diet, and so they can really change within the same subject. So I don't think it's a reliable biomarker unless you have extremely elevated concentrations. It might be that I didn't eat meat, I just ate vegetables the night before blood sampling, and the triglyceride will be lower in the morning if I'm fasting.
But we have not found, sorry, Roberto, we have not found the statistical significant differences between healthy controls and CCM patients.
Correct. Brava.
So it's not the main biomarker related to the inflammation. We have added this biomarker in our study so far, if I'm not wrong, Roberto, is just to relate this biomarker also to maybe some inflammation for the, already described for the cardiovascular disease, but also we aimed to correlate this data with the microbiome analysis. So this is
today the microbiome has been relatively not encouraging, but we have some way to go, and I hope.
Yeah, but we checked this biomarker for many reasons, but at this moment we have not.
I wanted to make a quick question to Favour. You described, and we all spoke about this increased inflammation, but do you have any dream or hypothesis of by which agent or event is this inflammation driven? What is the trigger? Because you see the shot of the gun, but you want to know which was the gun and who pulled it. So this is
a really good question.
Yeah, thank you so much for that question. That keeps me up sometimes. But so there's been some studies that have shown that when endothelial cells are activated, then they are able to trigger activation of immune cells. And so we already know that the loss of CCM3, or in, the CCM disease context, that endothelial cells become activated. And so that's definitely one pathway right, that could be causing it. And that's something that we are currently looking at, and we are hoping to publish in the coming months. But yeah, so that's one of the pathways that we think. So.
Thank you so much. And just as we're talking about this, I'm reminded to say to people, if you'd like to know more about the gut and microbiome, there's a webinar recorded from 2023 with Dr. Shelley Stevens on the Cavernoma Alliance UK YouTube channel on the gut and microbiome, and also on the Alliance to Cure website under lifestyle. There's quite a bit around microbiome and gut and the value of a healthy diet. So I'm just going to take, we've got 3 more questions and just one bit of feedback. So I'll quickly go through those. I'm just going to say this lovely comment. I don't have a question, but I just want to thank you all for your presentation. My 12-year-old daughter, CCM1, and she is the only Icelander to have this disease. This truly gives hope that one day there will be a cure. Thank you and greetings from Iceland. I mean, isn't that
wonderful,
incredible motivation to have? So I'll just take 3 more questions and then apologies, I'm going to close, but thank you ever so much, everyone, for your thoughtful questions. So this is about CCM3 and, where could you see more inflammation in CCM3? Anyone would like to talk about that and potential differences with that CCM?
Yes, so the question was more like why it's more, or?
And I think it's probably a question for those on this webinar who are not familiar with the differences, CCM3 presents a more severe form.
Exactly.
And I guess the question there might be about, would you be expecting to see more inflammation in CCM3?
Okay. This is sort of more of a guess, and if we can refer to what our mouse experimental model shows, the mice with CCM3 are more severely ill than CCM1 mice. But it's, of course, mouse, and if they would be kept for a long time, if that would be possible, maybe they would sort of team up in the long run. But CCM3 is also known to, on a molecular level, to be able to act in solitude and thereby affecting the vascular bed in a different way. And maybe that is one thing that might be a reason for CCM3 to be more severe than the other ones, and thereby driving, in this case, probably inflammation more than CCM1 would.
Thank you very much, Petra. So I'm just going to take the last question. I took these in order, so I'm so sorry, for those we won't be answering today. And this is a question about blood clots in a cavernoma. So for those who've been reading papers on aspirin, you may already know a little bit about this. So this may be one for Roberto. Thank you very much for your fantastic work. Can there be blood clots in a cavernoma that could cause symptoms?
Maybe I can answer. We believe definitely, because when those clots happen, then they essentially occlude the vessels. And so you definitely have hypoxia, which means you have less oxygen, going through, which means eventually you could have death of the other cells, right, in that area. But you can also imagine that when there's leakage of the components of the blood clot. So we know, for example, the red blood cells, if they leak and then when they get broken down, those products are actually toxic to the brain cells, and then those could also lead to some of the symptoms, and that has also been shown previously. So definitely yes.
And it's an interesting one with aspirin and blood thinners. Often people were told years ago not to take them, and there's more and more papers coming out actually about the potential benefits, which is a very interesting area. But just to reiterate, please don't go out and start taking aspirin or propanol. Please only act on medical advice in terms of any medication taken. So everybody, I just want to say a huge thank you to our presenters, and what a wonderful team effort from across Italy, Sweden. It's been wonderful having people from all over the world join us tonight. I know many more people will be watching the webinar on demand. So thank you. It's hopeful, it inspires us, and it may take time, but we're taking those steps forward, and with thanks to our presenters, we wouldn't be doing that. So thank you ever so much, everybody.
Thank you so
everyone.
much.
Thank you so much.
Thank you, everybody.
Bye.
So I'm
A
going to close
pleasure.